Assalamu alaikum, dear students. Welcome to online classes. I will try my best to make it convenient for you all. Today, our lecture is based on the construction and the screening of the DNA libraries. DNA libraries can be constructed and screened for genes and other sequences of interest. We can construct the DNA libraries, and after their construction we need to screen all those whether our gene of interest is being added in this library or not. This is very important to know about, uh, whether the gene of interest is added or not. There are almost there are some types of the DNA libraries. The important ones are the genomic DNA libraries and the cDNA library. And genomic DNA library is actually the set of the DNA clones which collectively contain the entire genome of an organism. This is the entire genome of an organism present in a DNA library. But for if we consider the cDNA doesn't contain the entire genome. It contains a region of the expressed genes, mostly synthesized from the RNA by using the process of the reverse transcription. Cloning reconstruction fragments with the complementary single-stranded ends. Uh, the procedure for this for this segment is: first we isolate the plasmid from the bacterial cell, then we cut that plasmid with the destruction enzymes to make the sticky ends. Simultaneously, we cut the gene of interest. Say, for example, in this diagram, we cut the gene of interest from the mouse genome, beta globin, and then we mix the plasmid and the gene of interest with each other. As previously we studied that the major aim of the DNA to get its own counterpart, but to some extent, to some regions, uh, DNA of one species can mix with the other, and making the hybrid of that particular genome. For example, this is the genome of the plasmid DNA. This is the plasmid DNA, and this is the cut segment of the mouse genome. When we mix both of these, after their mixing, we will get the hybrid and the recombinant DNA. We will get the hybrid DNA containing the plasmid and the insulin and the beta globin gene. The recombinant DNA contains the mouse EcoR1 restriction fragment inserted into self-replicating E. coli plasmid DNA. Amplification of the recombinant DNA. Antibiotic sensitive recipient cells are transformed with the recombinant DNA molecules. Transformed cells are selected by the growth under conditions requiring the presence of the selectable marker present on the recombinant DNA molecules. I already discussed this in the class that when we construct the library, after the library, after its construction, we need to know whether our gene of interest is added in this particular plasmid or not. For that purpose, we select the colonies of the bacterial cells, which can grow on a certain specific condition. Say, for example, with the insertion of the beta globulin gene, we may also insert a gene of antibiotic resistancy into the plasmid DNA. If bacteria grow in the antibiotic environment, in the antibiotic containing environment, it means our gene of interest is added. Synthesis of the double-stranded cDNA from the mRNA. The second procedure is the cDNA libraries. For making the cDNA, we need the RNA molecules. We need the RNA molecule. We isolate the mRNA and synthesize the DNA against that particular molecules. For this purpose, complementarity must be generated. Complementary strand is required to be generated. So, for example, this is the messenger RNA. This is the messenger RNA. But we use the reverse transcriptase enzymes and definitely the DNTPs. Uh, this reverse transcriptase enzyme generate the complementary strand against the mRNA against the complementary strand. Okay, so when this complementary strand will form against the messenger RNA, it will be formed cDNA mRNA complex. It is a duplex actually. It is the hybrid molecule containing the mRNA portion and one the DNA portion. So at the second step, we use the RNases to degrade the RNA. This procedure will degrade this messenger mRNA, and we use the DNA polymerase one for this purpose. This DNA polymerase one will generate the complementary strand against this messenger mRNA against this cDNA produced. So we also use DNA ligase. So if there is any nick to be sealed, after this procedure, the double-stranded cDNA will form. Screening DNA libraries for the gene of interest, genetic selection, searching for a DNA sequence that restore the wild-type phenotype to a mutant organism. Genetic selection, searching for a DNA sequence. We search DNA sequence that can store the wild type phenotype to a mutant organism. Genetic selection. We make the genetic selection for these organisms. Molecular hybridization is based on the hybridization of the similar DNA sequences. Colony hybridization. This procedure is to generate the colony hybridization. This is based on the 7th step. In the step 1, we synthesize the radioactive cDNA using the purified messenger mRNA. Using the purified messenger mRNA as a template. Plate bacteria carrying the recombinant DNA molecules on the agar medium and incubate until the small colonies are formed. First we create the radioactive messenger mRNA. Then we make the plate of bacteria. The clones of the bacteria carrying the recombinant plasmid. We insert our plasmid of interest into the bacteria uh, uh, in the bacterial cells. Now we replicate the plate colonies to a nylon membrane. Uh, if you know the replica plating, in this process we also make the replica. This is actually the process of the replica plating. In this process, I'm shortly explaining. In this process, first we synthesize the radioactive mRNA, then we insert it into the bacteria, then we insert the gene of interest into the bacteria. Now we need to know whether our bacteria is containing our gene of interest or not. We plate the bacteria on a specific medium. There will be some colonies grow on the plate. Now among these colonies, what type of colony is containing our gene of interest? We press this plate on a nylon membrane. We press this plate on a nylon membrane. Then we lyse the cell in situ, denature and bind the DNA to the membrane. We lyse the cell in a way that the rest of the cellular material will be washed out, and only DNA will remain on that particular place on the nylon membrane. Then we add the radioactive cDNA. We add the radioactive cDNA which we have synthesized in the talks. Radioactive cDNA and incubate under the annealing condition. At this condition, the radioactive cDNA will bind to its complementary part. It will bind to its complementary part. It will bind to its complementary part and rest of the DNA will remain at the place. Just the DNA will remain on the place. Okay. Now we washed out all the other DNAs which is not bounded in the nylon membrane, and only the binded DNA will remain on the place. Now we can find out where that DNA is being placed. We need to look it on the plate and find out that this bacterial colony is containing our gene of interest. Now we need to pick this colony containing this DNA sequence of interest from agar medium and isolate the recombinant DNA. Now, and we will isolate this recombinant DNA again to check whether it is our required DNA or not. Okay. This is all about the selection of the DNA libraries and how we make the DNA libraries. Now we are moving toward the molecular analysis of the DNA and RNA and the protein. For this purpose, for the molecular analysis of the DNA, RNA, and the protein, we use the agarose gel electrophoresis. We use the gel electrophoresis. We use the hybridization techniques. And there are many more procedures, but our focus in this class will be only on these procedures. Analysis of the DNA by the Southern hybridization, or we can call it this Southern blot hybridization. This technique was devised by the E.M. Southern in 1975. The DNA molecules can be separated by size by the gel electrophoresis. Then DNA molecule can then be transferred from the gel on the nitrocellulose membrane, just like the replica plating as you studied earlier. Uh, then DNA on the membrane can be hybridized with the DNA probe. Okay, this is the agarose gel electrophoresis. This is the procedure for the agarose gel electrophoresis. We prepare the semi-solid gel electrophoresis with the wells of the DNA sample. Okay. Then we place that particular gel into the running tank, and then we pour the sample into the gel, and then we provide the electric current then we provide the electric current to this agarose gel, and at the end we will have the image like this diagram. We will the DNA running will be complete after some time, and we use the ethidium bromide for making the DNA visible under the UV illumination. Okay, then what will we do? How is southern blotting will be done? Southern blotting is actually the transferring of the DNA from gel to the membrane to a membrane. In this process, this one is the glass baking dish. This is glass baking dish. In this dish, we will add the transferring solution. Solution you can call you can say buffer in this dish. Then upon this dish, uh, this is the glass plate. It will carry this glass plate will carry the our DNA, and this will carry our gel from where we need to transfer the DNA molecules. Upon that particular glass plate, long Botman filter paper sheets will be placed. Upon these long Botman filter paper sheets, nylon membrane sheet will be placed, and upon this nylon sheet, again we add the Whatman filter papers up to the 3 mm sheets, and it will be and these will be 3 to 4 sheets. And again upon this one, the 3 to 4 inches of the paper towel, and again the glass plate and the weight. Okay. Um, between these filter paper, nylon membrane, and the towels, we place the agarose gel electrophoresis. Buffer will move from filter paper to the agarose gel plate, agarose gel, through the process of the capillary action, and then move to the nylon membrane, and then move upward, then move to the nylon membrane. Okay. By this process, the replica of the DNA is generated on the nylon membrane, where we denature the DNA, sometimes before or sometimes after the transfer of the material. We denature the membrane by using the alkaline solution and then immobilize the DNA on the nitrocellulose membrane, where we hybridize the DNA with the DNA radioactive probes, and then we can find out our gene of interest by checking the hybridization of the DNA, by looking on the hybridization of the DNA, whether our DNA of interest is present in this particular fragment or not. Okay, this is the detection of wild type and the mutant alleles and alleles of the cystic fibrosis in a family. You can see the heterozygosity and the homozygosity for the analysis of the RNA by the northern blot hybridization. The northern blot procedure is nearly identical to the southern blotting. It's almost identical with the southern blotting. Both the procedures are the same. RNA is actually sensitive to degradation by the RNases containing with these enzymes must be prevented. So this procedure is actually sensitive for degradation, so it requires extreme care. So the RNA must not be degraded. RNA molecules contain extensive secondary structures and must be kept denatured during the electrophoresis. It contains the extensive secondary structures, so, and must be denatured under the electrophoresis, because the secondary structure will clear the problems, will clear the issues for our proper detection. Northern blots are useful in the studies of the gene expression. By using northern blot, we can study the expression of the gene. We can study how much the gene is being expressed. This is the image of the northern blot from the root, leaves, and flowers of the Etheliana. Okay, this is already discussed. I'm not going to. Analysis of the protein by the Western blot techniques. Polypeptides are separated by the polyacrylamide gel electrophoresis in the presence of the detergents that can denature the proteins. We use the polyacrylamide gel electrophoresis. This is also the type of the electrophoresis. Proteins are transferred from the gel to the nitrocellulose membrane, and individual proteins are detected with the antibodies. For the hybridization of the protein, we use the antibodies. Okay. Here you can see, say for example, this is the Western blot. We transfer the protein on the nitrocellulose membrane. Okay, after the transfer of protein, we treat it with the primary antibodies, uh, which only bind with the target proteins, the protein of interest, the protein for which we are looking for. Okay. After that, after the attachment of the primary antibody, we attach the secondary antibody. Uh, we attach a secondary antibody. Uh, the secondary antibody is is labeled with some sort of you can say the radioactive substance, and this will attach with the primary antibody, and where it will attach, it will show its signal from where we can detect that this is our protein of interest. Okay, this is all about the molecular techniques. The molecular analysis of the gene and the chromosome, the site at which the destruction enzymes cleave the DNA molecules, can be used to construct a physical map of the molecules. We can map the molecules. We can map the genes by using the destruction enzymes. However, nucleotide sequences provide the ultimate physical map of the DNA molecules. We can also use the DNA sequences. Okay. First, for the mapping, we use the restriction enzymes. If we use the restriction enzymes, we can check how much the two regions are far apart. Say, for example, this is a gene. This is the uncut nucleotide pairs containing the 6,000 nucleotides. This is the uncut fragment containing the 6,000 nucleotides. We obtain its image on the agarose gel electrophoresis. When we run this uncut fragment on the DNA, on the molecular gel electrophoresis, on the gel electrophoresis, it will give us the fragment near the near around 6,000 nucleotide pairs. Okay, here you can see this fragment is showing the 6,000 nucleotide pair. This one is the marker. This one is the marker. Okay, marker tell us the exact size of the DNA, how much the size of the DNA we are looking for. Okay, when we cut it with the EcoR1, when we cut this with the EcoR1, it breaks down the DNA fragment into the 2000 nucleotide pairs and the 4000 nucleotide pairs. 2000 nucleotide pairs and 4000 nucleotide pairs. And when we cut this fragment with the HIN3, it will cut the DNA into the 3000 and into the 5000 and the 1000 nucleotide pairs. Okay, here you can see in the line B, when we cut it with the eco R1, here the fragment of 4000 and here the fragment of 1000 being obtained. And when we use the HNC, we are obtaining the 5000 and we are obtaining the 1000 fragments of this particular DNA. But when we mix both equal 1 and HIN3, it will generate the cut among, so for example, uh, it will cut it into the 1000 and 3000 and the 2000. Okay. Why it is being happening? Because HIN3 is cutting into the 3000, HIN3 is cutting it into the 5000 and into the 1000, and hence he is cutting in between the region of 5000 and 1000. Okay, when we mix both of these, mix both of these, we will obtain three fragments of the DNA of the genome. One is the fragment of the 2000, second one is 3000, and again the last one is the 1000. And this is because the eco or one, why we are not obtaining the fragment of the 5000 fragments base pair, because the eco or one is cutting in between the 4000 fragments. So that's why 5000 cannot be obtained. Restriction maps reflect the true physical distances, unlike the genetic maps. Restriction maps give us the exact figure that how much distances between the two regions we are checking in the form of the nucleotide base pairs. How nucleotide base pairs region is far apart. The restriction map can be combined with other molecular techniques to construct the physical map of the entire genome. Sequencing of the DNA sequencing. Restriction enzymes, restriction enzymes to prepare the homologous sample of the DNA, of the specific segment of the chromosome. Gel electrophoresis procedure able to resolve the DNA fragments in length by single nucleotides. Gene cloning techniques allowing preparation of the large quantities of the DNA molecules. Sanger was the first who performed the molecular, who did the sequencing of the genomic DNA. Okay. A population of the DNA fragment is generated in the DNA sequencing. One end is common to all the fragments, and the other end terminates at a possible position. For this purpose, we use the dideoxynucleotide triphosphate. This is very important. We know that the deoxynucleotide triphosphate deoxynucleotide triphosphate contains 3' hydroxyl ends. But if we use the dideoxy, it means at the position of the 5' at the position of the 3' end, the oxygen is removed. Because of the removal of this oxygen, the DDNTPs, dideoxynucleotides, triphosphate, cannot proceed further for the elongation of elongation of the chain. So, for example, if you look in the DNA replication, you will know that the procedure required the 3' hydroxyl end. We know that DNA polymerase recognize the 3' free hydroxyl end for polymerization in the replication. But in the DDNDPs, this free 3' hydroxyl end is not present. So DNA polymerase cannot recognize this site and cannot proceed for further. Automated DNA sequences, now the fluorescent dyes are used for the detection of the DNA chains, and photocells detect the fluorescence of the dye as they pass through the gel or the capillary tubes, and the DNA sequence can be determined. For this sequence, for the sequencing, we use the template DNA strands. For example, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 fragments, 10 nucleotide phosphate we need to check, we need to sequence. We use the DNA polymerase, we use the primer strands having the 3' free hydroxyl N, and then we use the DDNTP. Against each DDNTP, we also use against each DNTPs, against each base, we also use its analog. We also use the DDNTPs. Say, for example, for DATP, we use DDATP. For DGTP, we use the DDGTP. For DTTP, we use the DDTTP. And similarly, for the DCTP, we use the DDCTP. Okay. Look on this we say, for example, we make the four test tubes. We make the four type of the reactions. One contain the DDG, one contain the DDA, and one contain the DDC, and one contain the DDT. Okay. In the tube where the DDG will bind, the reaction will stop. In the tube A, where the DDA will bind, the reaction will stop. In this tube, where the DDC will bind, the reaction will stop. And in this tube, where the DDT will bind, the reaction will stop. Okay. Now, by using this reaction, the fragment of different sizes will be generated. The fragments of different sizes will be generated. Among on the basis of those particular sizes, we can check the on the basis of the particular sizes, we can check whether our DNA of what we can check what is the sequence of that particular gene. We can check what is the sequence of that particular gene. So, for example, this is the A at the 10th position. On the 10th position, and here is the T. On the 8th position, again is the A, again here is the T. On the 7th position, again there is T, A, here will be the T. Okay, I'm going to number this at this position. This is 10, 10. This is 8, and this is, for example, 7. This is 1. This is 9. This is 4. Three. This is 3. This is 1, 2, 3, 4, 5, 6. This is 6. Okay, now we number it. At the position number 1 is T. 2 contains C. A. Again, at the 5 is A. And the G. This is the sequence that we obtain in different test tubes. Now we will generate its complementary. It's complementary. Complement. No, this complementary can be used for the larger fragments. We can use the different softwares. We can use online tools for making the recombinant of the sequence. Uh, we can call it the reverse complementary. So if you can look, this was our original sequence. While doing the sequence, we get this one. This one is actually the complementary of that particular strand, which is generated after the replication. Okay. The original sequence for which we are looking for was the AGTCs. No, we get the sequence of that particular genome. By advanced techniques, we can also get the nature products and separate by the polyacrylamide to the well electrophoresis. And sometimes we know this technique is automated. We use the fluorescent dyes, and that dye can be detected by the laser and the fluorescent detector, laser light and the fluorescence detector, and the data can be recorded into the PC and on the computer, and data can be recorded on the computer. We can get the data by using the DNA sequencing tools. So this is the diagram of the, you can say this is the print out of the DNA sequencing that we can perform on the DNA sequencer. This is all about the techniques. This is all about the techniques. Tomorrow we will learn about what are the social responsibilities of the recombinant by using the recombinant DNA technologies. Uh, Thanks a lot for watching this, and I hope um, it will be easy to understand. And I'm hoping you will have the question you will contact me. Thank you.